So I'm Harold Schwartz, and I'm very, very pleased to be able to do the introductions for these talks. I think this is a very exciting series, and I think O2M is making a very important contribution to the community. And I would encourage you to let your colleagues know so that they can take advantage of this service. So the seminars are a series of eminent speakers on various subjects. The one exception being the picture that's in front of you, which is an obligatory inclusion of somebody else, but I can assure you the rest are distinguished. And the host is the company, and that's the CEO, who will also be talking to us in March, and very important on encapsulation. In February, so we're going backwards, Carl Simon from NIST will be speaking, and in January, we have two speakers. The first is Boris, the other part, and Greg Palmer. And but now in December, as a part of a holiday treat, we have Howie Halpern. So Howie is a legend in his own time in the area of in vivo EPR and EPR imaging. Howie started life as a physicist and a high energy physicist, and then later went to medical school. And so he has brought to the field a rigor in physics and a perspective that is absolutely outstanding. And he's now supplemented that with his experience in day-to-day activities as a radiation oncologist. So we have the rare treat of someone who understands the physics and understands the applications simultaneously. He's done some pioneering work, trying to stop me. He's done some pioneering work in the clinical applications and in animal studies, and he really understands both the potential and the challenges. And so Howie, with no further ado, I'm looking forward to hearing your talk. So I'm pleased to introduce Howard Halpern.
So I'm going to try to share my screen. And Marcello, if you could allow the sharing of the screen, and there it is, and here it is. Okay, let's go to the beginning, and let's begin. Okay, so thank you very much, Hal. Again, I'm Howard Halpern, and I want to tell you a little bit about the possibility of enhancing radiation therapy, enhancing the therapeutic ratio of radiation therapy in general by targeting resistant regions of a tumor with EPR molecular oxygen imaging. And I've got a little conflict of interest with a couple of patents, and I'm a member of US O2M. And I want to start by saying that the goal of all medical intervention is enhance something called the quantitative therapeutic ratio or therapeutic index. And one of its definitions is when we give any kind of therapy, even the extent of surgery is in a sense a dose. And so that, and obviously every medicines that we give is a dose. And the therapeutic ratio is a dose for the 50% chance of unwanted therapeutic complications ratioed with the dose for 50% chance for the proper therapeutic effect. And the therapeutic ratio is the the ratio of those doses. So we want the dose for the bad effect to be large, the dose for the good effect to be small, and therefore maximizing that ratio, dx to dy, is the goal of virtually everything we do in medicine. We characterize it often as something that has a sigmoidal um, uh, appearance as a function of the dose we're talking about, also called the logistic function, where at low dose there's minimal response in both cases, and at higher doses there are maximal responses with probabilities of 1, so that we have something as indicated in this diagram from an article by a guy named Sam Hellman, where you have tumor control at a 50% level at a much lower dose than a major complication from whatever we're doing. And again, that's what I'm talking about. Now, I'm going to flip now to the nature of cancer. They develop from our own tissues, and they share much, maybe even most genetic identity with the normal tissues. They are, however, cells that have escaped normal growth controls, and as a result, they become a chaotic growth. As such, they are also spatially heterogeneous, and their sensitivity and resistance, by good evidence, is also heterogeneous. And the question then is, can we increase the therapeutic ratio with a spatially selective dose delivery? And can we then define something like sensitivity or resistance as a function of location in the tumor to show both improved tumor control and at the same time possibly reduced side effects? Hypoxia, the low molecular oxygen in a tumor or in normal tissue, in fact, all living systems, has been known for over 110 years to be resistant to radiation therapy in regions of low molecular oxygen concentration, as was demonstrated by another Schwartz, Gottwald Schwartz in Germany, when he bound a piece of radium to his hand and had a loosely fitting piece of radium, not to his hand, but to his wrist, and found that there was much more of an effect in the loosely bound relative to the poorly bound. Tomlinson and Gray in 1955 published papers showing that there was about a 70 micron radius, 150 micron diameter between tissue death and normal tissue, wherein tumor cords were found. And it was consistent with something called consistent with the diffusion length before the absorption of oxygen by rapidly metabolizing tissues, the Crow cylinder. You can see that from microelectrode measurements of a breast tumor, shown in figure A here with the arrow, the molecular oxygen values of a breast cancer are much, much lower than on average the molecular concentration of oxygen from normal tissue. In fact, using an Eppendorf microelectrode, when there was hypoxia, radiation control in cervical cancers here from Huckel and Files, from sarcoma, from Dave Brazel, head and neck cancers from Nordsmark and again from Dave Brazel, show that there is a doubling of the control of these tumors among tumors, that very, very significant tumors, when measured with a needle electrode, if the tumor is well oxygenated relative to those that are poorly so. the idea behind electron paramagnetic resonance imaging is that we need to defeat confounding variation in the measurement of the molecular oxygen using electron resonance. Animal physiology, human physiology helps regulate that. Temperature, osmolarity, viscosity, and pH are generally relatively well controlled to within small variations. But the idea behind this whole effort is to infuse a spin probe with unpaired electrons. These unpaired electrons are going to function like needle electrodes. If they're prepared in a magnetic field, the energy structure of these unpaired electrons on hopefully benign molecules creates a net magnetization, with the higher energy electron spins having a smaller population than the lower energy electron spins. And that depends on the sign of the charge, electrons, of course, being negative, protons being positive. But at any rate, that magnetization means that when you expose those electrons to an electromagnetic field with a frequency nu, that energy difference is increased at higher magnetic fields. And there is a point then when you have an agreement between the energy splitting and the frequency of your exciting field relative to the magnetic field B naught here, where there is an increase in the absorption of that energy. And that process is the electron resonance. Now, there are two ways in which to acquire the information about the environment, the oxygen environment of a spin. And one is to very gently sweep the magnetic field. And when the frequency of that magnetic field is equal to the resonance condition, there is an increase, an abrupt increase in the absorption of energy into the spin system. Or the second way in which we can observe things is to hit the spin system with a pulse of energy with a very large bandwidth that covers the entire frequency spectrum, and watch the electrons that have been forced into the higher energy state relax. And the relaxation rate is the same units as the change in frequency in a swept magnetic field or swept frequency system. And so that it basically tells you the same thing, the same information. They're related literally by a Fourier transform. Now, oxygen is a diatomic molecule which happens to have two unpaired spins. Like a bad marriage, the spins are stuck together in the same molecule. And Pauli's principle tells you that electrons cannot be in the same state at the same time in the same place, which means they're relaxing each other intrinsically within the oxygen molecule. The fact that the molecule is a rapidly rotating diatomic molecule in the magnetic field increases the relaxation rate, and the states there last in femtoseconds, which are too short to measure directly. So what we need to do is infuse a benign, well-behaved unpaired electron in a spin probe, which I show here is this P dot. And this long-lived spin state, and in the case of electron resonance, 7 microseconds at a very low molecular oxygen concentration is a very long time. And when that well-behaved electron collides with a an oxygen molecule bearing 2 unpaired electrons. This very briefly increases the well-defined probe relaxation rate through a process called Heisenberg spin exchange between the probe and the molecular oxygen. And we cannot distinguish whether or not the electron is located either on the probe or on the oxygen, and so that we have to describe it as a linear combination of the two, and therefore the probe electron for briefly takes the relaxation characteristics of the oxygen. Now, we can, because it is a magnetic resonance experiment, we can image it in preclinical models. We can do it in a normal adjacent tissue. We can do it quantitatively, and the quantitatively means we can repeat and believe the repeated measurements like a PO2 electrode. And the other thing is that we would like to make other confounding effects less significant, which, as I had noted, is provided by the homeostasis of living tissues. In the process, we've discovered that pulse spin lattice relaxation, which is not aligned with or not entirely aligned with dependent measurement, is far freer of the major confounding variation, which is the effect of the spin probe itself relaxing other spin probes. And the reason for this is that in spin lattice relaxation, the energy of the spin system of the probe has to be communicated with the surrounding lattice, and remarkably, oxygen appears to be the major lattice in a living system, whereas the line width, the spin-spin phase encoded measurements can't distinguish the probe-probe interaction from the probe oxygen interaction. And that's indicated here in this picture where we have spin lattice relaxation. You're insensitive to the exchange of energy between the spin probes because the relaxation is not affected without some leakage into the lattice, whereas in the spin-spin relaxation process, you are in fact equally affected by spin probes interacting with each other and oxygens. Maybe not quite equally, but at very low oxygen concentrations, the probe of your spin system from the probe is at least that of the local oxygen. So if you can identify hypoxia resistance, then you have the possibility of a radiation boost, which is provided now with a wonderful technology enhancement by something called Intensity Modulated Radiation Therapy. It's a general rule among radiation oncologists that you need to keep the dose, and this is a huge tumor. You can see it having eaten away at this patient's jaw, most of the jaw. And yet, if you have heterogeneities in its dose, you run the risk of necrosing bone or soft tissue in the radiation field. If some of the regions of that tumor are resistant, for example, those that are hypoxic, then you may be able to use a boost, avoiding the bone that is otherwise likely to die at the high dose hotspots or increased dose points. Now, the hope has been attempted with positron emission tomography of a nitroimizole, fluoromycinidazole. And here is a study from a French group, and the difference between those patients who got, and that's in this green line, who got a boost greater than the standard tumor dose of 66 gray, at least the hypoxia identified by the PET was identical, P of 0.5 difference between that and the F-MISO positive, those that had hypoxia. In fact, not only were the tumors not controlled, but the complications were about the same for the boost, indicating possibly that when you obey dose constraints, you actually are likely avoiding some of those critical structures which can affect quality of life. So that despite the sensitivity of the century of hypoxic resistance, no data from mammalian tumors, and there have been hyperbaric oxygen trials, an enormous amount of attempts at trying to enhance radiation. But the problem has been is that it's not been able, we have not been able to identify all of the hypoxic regions. And again, the chaotic tumor vessel delivery of PET tracer, low tumor extracellular pH, all of these may be confounding the identification of hypoxia using PET. So that the idea is to try to develop first preclinical and perhaps eventually clinical hypoxia images that PET cannot in itself do. Now, you can image by rotating gradients about the direction of a gradient in a magnetic field about your object, and that's what's done in computed tomography. This I'm going to go through quickly. You can even do a spectral spatial image using these rotating gradients. But unfortunately, most of the imaging systems that are used for the beautiful MRI images operate at much higher magnetic fields and frequencies, period, to enhance signal to noise. Standard electron resonance frequencies are 2 to 3 orders of magnitude greater, 10 to 300 gigahertz. But I 40 years ago almost, I decided I would try working with very low magnetic fields, 90 Gauss at about 250 megahertz, and more recently a little bit higher at 720 megahertz, which is the 250 is the frequency of a 6.25 Tesla MRI. By lowering the frequency, you increase the wavelength, you allow deeper penetration, and so that deep in what we call lossy tissues, we can see resonance processes taking place. It turns out that there was a much less than commonly expected loss of this signal to noise, and allows us to get images in at least preclinical models. We use this spin lattice relaxation. It involves a pulse, and these pulses are typically 60 to 90 nanoseconds long. They invert the magnetization, and then we watch as a function of time after that inversion, the magnetization returning to its normal state, and that's as the spins in the inverted system donate their energy back to the oxygen lattice. The signature of low and high molecular oxygen concentration after this electromagnetic pulse in low oxygen as a logarithmic function of linear time, you can see a very small slope. At high oxygen concentration, the logarithmic signal is a much sharper slope. And this is sort of the experiment on a mouse. Here's our animal inside in a cradle. Its leg bearing a tumor is inside a very simple resonance structure called a loop gap resonator, bladder flushing, a spin probe called a tridal, which is indicated here, a 1500 Dalton or so molecule with a single unpaired electron decorated with with no spin, essentially carbons and sulfurs, up out to 6 bonds from the unpaired electron, provide a narrow high signal that is very good at communicating oxygen. At 720 megahertz, we opined that it would be very convenient to confine our measurement system to a box 2 feet by 2 feet, which is the O2M system, the Vivo O2M system. Here's another one of those fairly simple loop gap resonators, which makes it with a 19 millimeter diameter. We can make them large enough so that they can accommodate at 2 inches large pieces of anatomy of even a rabbit. At any rate, when we inject the spin probe and we use the spin lattice relaxation and compare the oxygen sensitivity, as we see here, the graph is shifted because there's a little bit of an offset of the spin probe concentration between spin lattice and spin relaxation. But the slope is a beautifully linear, and that's actually the signal to noise in those dots, function of oxygen. Whereas as a function of spin probe concentration, there's almost no effect on the relaxation rate, a little one, but well within errors from the spin lattice relaxation, where there's an artificial increase in the oxygen, the apparent oxygen concentration reflected in the relaxation rate with the spin or relaxation, and it's about an order of magnitude increase. This is what a typical PO2 image looks like. Where here the color bar quantitatively relates the molecular oxygen concentration to the level of hypoxia in this 3-dimensional tumor whose boundaries are indicated here as this red contour, and that boundary comes from a T2 weighted MRI of the tumor. And you can see this one is an enormously hypoxic tumor with much of it well below 10 torr. And if you can recall back to those previous figures, that means that it's going to be very resistant to radiation therapy. Now, we did learn in all of this process that we need to be able to involve multimodality images, the MRI to define boundaries of the tumor, other images to reflect other aspects of physiology that would include the pH and again the PET signal, which is something that can be used in the clinic. And it argues that we need something with which to register these images, and we do put something called a fiducial, which is a fixed tube of a fluid or something with contrast that is sensed by all these images so that we can register the data sets. Now, there's another breakthrough, this IMRT. This is really not IMRT that I'll be talking about. It's conformal radiation therapy to mice with a gantry-based X-ray with very low, but again, only animals, energy. And we did an initial experiment wherein we treated the whole tumor to either a 15% for the sarcomas or a 20% control dose, and looked at that as a function of radiation dose for control of the whole tumor. Here's the 15% control dose at 22.5 gray. Here's 95% control at 35.5 gray. That's the radiation dose unit. And we tried at first to use just crude spherical volumes or shells to avoid, to treat the hypoxia, or to just treat an annulus around the hypoxia. This is what we got. And it was total depression. There was no difference, a p-value of nearly exactly 1. And the radiobiologist, of course, told us when you have only 85% of the hypoxic voxels or clonogens, they're going to grow back. You're not going to be able to control them. So that at that time, it was very clear that it would be possible using 3D printing to identify 98% or more of the hypoxia and direct a conformal boost with these rapidly printed tungsten-loaded plastic blocks. Now this is not hypoxia you can see in this plane, but it's an out-of-plane hypoxia projected onto the plane perpendicular to the beam direction. And here is the major hypoxic volume. But now you can make a 3D model of it. And then here's the printed block wherein you don't quite get the isolation of the island and it becomes a peninsula, but that's okay because we really need an extra millimeter, 1.2 millimeters to take care of setup variation. But this is the second set of experiments that we did. Here, this fine line here is all of the hypoxia, the fine magenta lines throughout the tumor projected onto this central plane. Here, and here's another dot of hypoxia, and it's projected onto the central plane, and they're given a 1.2 millimeter margin for the hypoxic boost, or we randomized to a treatment that blocks the hypoxia and erects a treatment volume for the boost of roughly, certainly equal area and roughly equal integral radiation dose volume. And we boost to a 95% control dose for the sarcomas, to a 99% for a breast adenocarcinoma. And this is what we got. And this was a very satisfying increase, doubling of the control, somewhat similar to what you see from the original Eppendorf electrode values when you boost the hypoxia relative to if you boost the well-oxygenated tumor. And this is repeated in a breast adenocarcinoma treated to 20% of the control dose, and then boosted with a 99% control dose. And you can see it turns out to be even more significantly different. You can also see that the first animals in this group gave us a little bad news. Now, we've got a similar experiment that we're doing with the GIVA-25 produced by O2M, and lo and behold, first 8 animals, the first one to fail was a tumor hypoxia boost. But if we go back and look up here, the first tumor in this group to fail was a well-oxygenated tumor. So early in the comparison, but it looks like we're getting similar kinds of data from Jiva as we got from our first experiments on 2 tumors. This is the first confirmation in mammalian tumor study for 110 years of the effectiveness of boosting these hypoxic regions, the radiation-resistant hypoxic tumor regions. Is it the only source of resistance? Certainly not, at least, but it's certainly a major one, maybe the major one. And the idea here is that we know that PET has problems in imaging hypoxia, maybe due to the fact that instead of looking at the behavior of your probe once it gets to the hypoxic region, all PET does is that it monitors the amount of probe that gets to the hypoxic regions and binds there. That then may mean that the vasculature is inadequate and doesn't adequately reflect the hypoxia. So we got a grant to do some imaging of PET scans registered with EPR scans. And the idea was that to look at the diffusion of F-misonidazole and trittle together in a set of experiments. And we used dynamic contrast enhanced MRI with gadolinium to look at the tumor vasculature. Eventually, we're going to do some iopamidol CEST. And the hypothesis is that we can combine information about hypoxia from clinically available imaging, PET and dynamic contrast enhanced MRI, with the PT, the EPR PO2 as a ground truth, and use the EPR to tell us how to correct the PET so that it is providing a true oxygen imaging. And the first experiments, we use an EPR with the blue boundary or the magenta boundary showing hypoxia, a T2 MRI showing the boundaries of the tumor, dynamic contrast enhanced K-trans and VE, and then PET. And we have them all obtained together, model the retention of the PET tracer, again, as a logistic function, okay? And then try to reconstruct our EPR image through this logistic function model to look like a PET, which we're calling PET hat here, and then correct the PET hat or the normal PET with the dynamic, just the intensity of the voxels and a dynamic contrast enhanced MRI to better reflect the PO2 of the cancers. And it's again, obviously, we can see that the agreement between, this is the distance between the boundaries of a PET tumor to muscle ratio, 1.4, and a 10 torr hypoxic volume in an EPR. And obviously, we're massively increasing the overlap with a simple linear correction based on the dynamic contrast enhanced voxels. Now, again, that may be very primitive, nonlinear corrections, addition of this pH image, iopamidol CEST, which we're apparently making some major progress with, is occurring. We've got now a PET scanner for which we've been granted some money that sits inside a magnet identical to that used for the Jiva And we're able now to obtain our PET scans. Here's the EPR resonator. Here's the PET for the animal leg-borne tumor, avoiding delays. And we've got a new group of images that appears to show a similar improvement from just the dynamic contrast enhanced radiation. All right, I think I have another few minutes, so I'm going to the possibility that we can do this in people. One, I think that it's important to use high signal to noise bimodal surface coil antennae to excite and detect the excited spins and do our spin lattice relaxation. And this is a model of one of those. I want to remind you that this very simple model of the human body is a set of simply connected tubes through which an endoscope, a flexible endoscope, can be threaded to, and then have the resonant structure on the surface of a balloon that can be blown up to enhance or encompass even a non isolated tumor. Maybe just one half of the tumors has the resonator wrapped around it. But that gives us access to esophagus, stomach, essentially all of the colonic cancers, uterine cervical malignancies, head neck malignancies, a little more struggle with lung cancer, which is a major killer. But at any rate, if we can do those local images with surface coils, and this is the model, this is the actual surface coil, and as you can see, the signal from a loop is bigger, but it disappears into the noise compared to the what we call the butterfly coil, and you can see significant oscillations as the magnetization dies off. And at any rate, if you could do also local injection of the tube of the probe, you can get a 4 order of magnitude reduction in the probe, and you can see here that local injection indicated here in the very low oxygen areas very nicely reflects the actual intravenous probe measurement, so that it might be possible, and in fact, it may work in humans, like as indicated here. And a little calculation shows that what is currently injected into people using a metabolic carbon-13 hyperpolarized pyruvate metabolic image polarized with the same spin probe that we use for oximetry, the dose of the spin probe that gets into people is 1 order of magnitude more than the dose that would be that is given to our animals, who seem to be tolerating the spin probe very well. So that this is the basis for some optimism that we might be able to do this in people. In conclusion, we've gotten some quantitative oxygen biology. The spin lattice relaxation based PO2 images gives us about a torr accuracy and about a millimeter spatial resolution. And it's given us the first data indicating clonogenic control in mammals based on a 10 to our hypoxic fraction is reasonable. It may help define some of the corrections that can be given to PET, but I think that the direct measurement of the oxygen is likely to be eventually better, and that small animal information can help inform human cancer treatment, and the EPR images may be possible in human subjects to improve the cancer therapeutic ratio. And these are some of the people who were involved in those experiments. And there are groups from radiology at University of Chicago, colleagues from the Novosibirsk Institute for Organic Chemistry to provide Tridal as well as General Electric, and then of course our city of Chicago, to which I would love to invite those who are not here to visit once this pandemic eventually leaves us. Thank you. Thank you for listening.
Thank you, Howard. Please
Did anybody
unmute
hear that?
yourself for any questions you have.
Well, I have a few. Can you hear me? This is Michael Garwood.
Hi, Mike.
Very nice talk, Howard. Thank you very much. I'm very interested in this technology, also not just for cancer, but for measuring PO2 in a bioartificial pancreas. You know, I've got a collaboration with Claire Coates-Pappas down at University of Arizona. And we've been using fluorine and, you know, NMR and T1 of the fluorine, but this looks pretty interesting. So I've been making a little foray into EPR myself. And I think the way to go is at very low field, and what you're doing is really cool. That's bimodal surface coil was very interesting. What is it? So why did you, I didn't quite get why, is that for MRI and doing EPR? Why was it so fancy?
Well, I mean, the idea of isolating, and the current paths involved in the excitation pulse are orthogonal to the current paths of the detection side of that resonator. Okay, and you, as I don't know, I glossed over it quickly, but we got near 40 dB isolation between the excitation and the detection system. And because of that, you get a much cleaner detected pulse. And okay, that was, and in fact, it was with you guys that we designed that. And the first version of it, in fact, the one that's indicated there, was built in Minnesota. So, you know, that's Tommy Vaughn's group.
Okay, understood. That's one of the the leakage problem, but you're doing pulsed NMR, so you can't switch fast enough. You're basically describing the leakage of the transmit to the receiver.
Oh yeah,
that's why
yeah.
you need that 40 dB.
Yeah, exactly.
Yeah.
I mean, you know, this is a 60, 70 nanosecond pulse, and it blows away the first several hundred nanoseconds of your detection system. So you lose a lot of signal in that process.
Yeah.
It's not, you know, even solid state NMR isn't quite that quick.
Totally understand the problem. Been working on it, and actually, look in JMR this month. We published a paper where we longitudinally detected EPR and got 80 dB.
80 dB. Oh wow.
You could simultaneous transmit and receive has been a problem that I've been after for years with Tommy Vaughn and others. And anyway, this is very nice. I'll let somebody else ask some questions.
Sure.
Hello, everyone. This is Martyna.
Ah,
It was very nice
hello.
to hear the talk. I have more general question that comes back to some of our previous discussions, Howie. So I think the increase, factor of 2 increase in radiotherapy effect is very good. So, but do you think is it possible to get even higher? Is it the issue with the size of the tumor or the animal models we are using?
Look, that's a wonderful question, Martina. And one of the things that is in a way intellectually problematic about our approach is that the effects of radiation are supposed to be primarily double-stranded breaks in the genetic material, and that's an internuclear problem. Our probe as a triacid, a 1500 Dalton triacid, is only sampling the extracellular portions of the tumor. And the fact that well, of course, that's what's being sampled in the needle electrode measurements. Okay, so that it's not surprising that that aspect of it is recapitulated. But it would be a very interesting experiment if one could provide some means by which you could convey that tridal into first the cell and then eventually to the nucleus to see whether or not the hypoxia and the definition of the hypoxia might be improved by intracellular measurements and intranuclear measurements. And there are we've got a paper that we published showing that there is internalization of spin probe in mesoporous silicon nanotube structures, and that the preliminary data on that shows a slightly lower tumor oxygenation, and in fact there may be active transport mechanisms between cytosol and nucleus that can be exploited. There are designs using a single nanotube that might allow the tridal to be transported through the nuclear pore into the nucleus where the double strand break mechanism is taking place. So that there's a whole lot of biology that's in front of us that is needed to answer those questions. And there are means by which we may be able to do that.
Yes, absolutely. That sounds fascinating.
Hey, this is Carl Simon from NIST. I had a question about the human imaging. So do you think you can give a patient trittle at a high enough concentration to image and that it won't be toxic to the patient? Thank you.
That's a great question. I do, and the way in which that would be done would be as you introduce the surface resonator through the endoscope. You can also introduce an array of localized needles that would locally inject that spin probe at that 4 orders of magnitude reduced dose. That I don't know if it was clear, but it looks like a couple of hundred micromolar concentration in the tumor gives us these very nice distinctive results. And by doing that local tumor injection, and for example, and this was with a T2 sized tumor, which is in most staging systems a 2 to 4 centimeter diameter tumor. That's not a small tumor. Again, about a 15-20 microliter, milliliter tumor. and that would give you a very low burden of trittle in a patient. So, I mean, and the point is that it's already going into people at 1/10 the concentration necessary to do that. So the issue is and I'm unaware of, again, there's been several hundred people that have been so treated at Memorial Sloan Kettering, and I think Hopkins has got a polarizer. Mike, do you have a polarizer in Minnesota?
Yeah, it's a hypersense, but I don't think it's working right now.
Yeah, no. I mean, they are complicated. That's
Yeah.
right. Yeah, I mean, it's slowly becoming apparent that there's a real use to these things, and we just need to understand that biology. So yeah, I think it can be.
Products being developed that might be less toxic. And my other question was from talking with Marignani that the trittle can be hard to get a hold of. Is that situation changing? Is there?
It is. There are people who are finally discovering it was a trade secret that for the synthesis, and there's a final step that requires some real art in making sure that you've got what is a really stable molecule. But the group at West Virginia have published on a means by which to make the simpler versions of that, and certainly now the tri-alcoholic versions of it. It's the alcohol that makes it very much less toxic, and that is and that looks like it's doable at more than just a couple of places.
Howard, I wanted to just add that we also hope to do it at O2M. That's why I put up the position announcement for O2M. So we hope we'll solve this problem.
Yep, that's right. Yeah, no, and it looks like it's some flow technology and the ability to do that at low temperatures to prevent the thing from falling apart just at the end of the synthesis. It involved synthesis, but and the group in Siberia, that's where I went, which actually it's true. In the wintertime, it's very dry and very cold, and that's what's necessary to do this. It can be done without that. And the group, the GE group that invented it, can make it in kilogram quantities.
Thank you.
This is Hal Swartz. Very good. Howie, lots of really exciting things. It's just amazing. I'd like to get your thoughts, particularly on the possibility, whether it's in animals at first, of getting the measurements intracellular.
Yeah.
When you think about radiation biology, it's the thing that kills cells in spite of everything else, and other things that factor is DNA damage.
Right.
And so the only oxygen concentration that is important is the intranuclear oxygen. And at least getting intracellular oxygen rather than tissue oxygen would be just a game changer in terms of understanding. We've been talking at Dartmouth about trying to understand flash.
Yeah.
And flash seems to be oxygen dependent, but it can't be the tissue oxygen. It's got to be the oxygen at the nucleus. And in that moment when you have very high LET, and to be able to, one, calculate, but also to measure it would really be exciting.
Yeah. No, well, we're trying to, we're trying, using the technique that we published, to do that. And that is in the works for us to try to go after that intranuclear and certainly intracellular molecular oxygen concentration. That's a good point, Al.
Are there any more questions?
Hi, Howard.
Hi, Molly.
So I have only one question about those blocks that are used to cover the radiation.
Yep.
Is it something possible within the treatment time, like within few hours, or it takes a day?
Oh, no. It takes, I mean, the apertures take 10 minutes.
10 minutes, oh, that's very
I
fast.
mean, using a 3D printer. I mean, these are relatively small tumors and so forth. So 10 minutes per block. And so that does introduce a little bit of a time delay between the actual oxygen measurement and the boost treatment. But that's often going on. All the registration and the printing and so forth during the whole tumor radiation. Look, we have we're working also on real intensity modulated radiation instead of using these conformal apertures, using a compensator based delivery, and it looks like 3 fields rather for most of the tumors that we've seen in these animals is about enough. And that, if you know, again, the compensators don't require quite as much, particularly if you can surround the actual compensator, for example, made out of a copper plastic with a tungsten loaded sleeve to prevent some of the edge scattering. We can make those relatively quickly, so that shouldn't delay things that much.
That's
I mean,
great.
part of the problem with our technique is the it's been a learning curve using, and I think they've learned with us using the isocentric delivery system, the XRAD 225A system, and that which it comes from Princess Margaret Hospital, and made in the U.S. But at any rate, we've learned with them, and they've software issues of gantry accuracy, all of that are really tough medical physics problems, and we've kind of come by that. I mean, the last year or so has been remarkably free of some of the glitches that have occurred in that system. And again, it's been 10 years that we've been using it, and now we know.
It's amazing that so you can do it in real time almost.
Yeah.
Hi, this is Greta. I was wondering what other factors you said that make the tumor resistant to treatment besides the hypoxia. I was just wondering.
Well, I mean, the hypoxia isn't necessarily stable. I mean, it is a dynamic system, okay? And there on my screen is Greg Palmer from Duke, and Duke provided some of that hypoxia information in those first slides that I showed. And they also know through work by Mark Dewhurst about the transient component of oxygenation. It's a little controversial what fraction of tumor hypoxia in humans is and what of the transient hypoxia relative to the dynamic hypoxia, but it changes. So that's one of the sources. And of course, there are going to be other physiologic and molecular biologic sources of resistance to radiation therapy.
Maybe pH and redox potential or
redox, all of those. Yeah, that's right. And in fact, that we are in the process of imaging with a pH sensitive image to throw into the mix of correcting the PET, and that I'm sure we've got some really wonderful collaborators in that process. I mean, it's there really are a lot of good people in this area, and at any rate, and it sounds, it looks like there's an enormous amount to learn still about dealing with the tumor microenvironment. I mean, certainly measurable stuff is important, and biochemical solutions. There may be a means by which to better oxygenate tumors using clinically available chemicals that are used to treat porphyria, which has been around since King George III, and basically whose disease may have helped the United States give birth to itself. So there's again a lot of stuff around.
Thank you.
Yeah.
But it's kind of counterintuitive for me, actually, because from my understanding, tumor has a lot of vasculature. So since from what I learned, blood carries a lot of oxygen through our system, it's counterintuitive that a tumor with lots of blood vessels would have less oxygen.
But the issue is it's a chaotic vasculature, and there's a lot of work that's been done just looking at casts of the blood vessels, which you can do by just infusing some crazy glue, different ways to assess the vasculature, and that vasculature is not an effective deliverer of both oxygen and nutrient to the tumor, so that the tumors have an enormous overturn in their cells. The ability of an animal system to in an organized way to renew itself is amazing. And that's the advantage that we've been exploiting in radiation therapy, particularly giving the patient system an opportunity to heal, at least partially heal, in competition with cancers, which are such chaotic organizations that they can't begin to do what the normal renewal systems are capable of doing. And that really is, that I think answers the questions of all of this vascular, tumor vasculature synthesis. Yeah, you're right, there are vessels there, but they don't work very well.
Thanks. And one last question: when you mentioned about the nucleus and trial molecule,
Yep.
does the membrane have to be ruptured for the trial to go inside the cell, or
No.
it doesn't need to be?
No, it doesn't. These nanomaterials are, you can paper the outside of these nanotube structures with some proteins that stimulate endocytosis, and so that it's a natural internalization that takes place.
I see. Thank you.
And you can actually synthesize proteins for the surfaces. Mitochondria would target rough and smooth endoplasmic reticulum. I mean, the whole, you can go do an oxygen biology of the cell these days in an animal. And that's kind of amazing too.
Yeah, I just wanted to make a comment about Mike Garwood's comment. So yeah, we are looking at the bioartificial pancreas, and also Clear Host Papas also is part of the consortium. We started at O2M. It's called Oxygen Measurement Core, and we are looking some of those sample with Jiva 25. So thank you for making that connection.
Yep, great.
If there are no more questions, I would just like to thank all of you for joining, and I hope to see you again in January when Greg Palmer from Duke University will present on oxygen biology, radiation biology experiments, some of the results and what they're doing with oxygen measurement, as well as biology of the cancer. And thank you for joining, and please spread the word about positions. We're looking for good people to join our company. Thank you. Have a great week.
Goodbye.
Thank you.
Bye-bye.
Bye-bye.
Bye. Bye, Howard.
Thanks, Marianne.
Bye-bye. Goodbye, everybody.
Bye, Dr. Swatch.
Bye-bye.
Great.